Basics and Beyond. Chapter 16, the Major Histocompatibility Complex or MHC. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we explored the complement system and its vital role in the immune response. Today, we will turn our attention to the Major Histocompatibility Complex or MHC, a group of molecules that are central to the adaptive immune system's ability to recognize and respond to pathogens. I briefly introduced the MHCs in a previous chapter. However, its importance necessitates a separate chapter. So let's talk science. To introduce the MHC or the Major Histocompatibility Complex, it is a set of cell surface receptors that are essential for the immune system to recognize foreign molecules. These proteins are found on the surface of almost all nucleated cells and plays a critical role in antigen presentation, the process by which cells display fragments of proteins, called antigens, to T cells. This interaction is crucial for the activation of T cells and the initiation of the adaptive immune response. There are two main classes of MHC molecules, MHC class I and MHC class II, each serving different roles in the immune response. Before we dive into the specifics of MHC class I and II, let's briefly touch on how antigen processing and presentation work. Inside cells, proteins, whether from the body's own cells or invaders like pathogens, are continuously broken down into small fragments called peptides. These peptides are loaded onto MHC molecules and transported to the cell surface, where they are displayed. This process allows T cells to scan for signs of infection or abnormalities. The MHC peptide complex acts as a flag, signaling to T cells whether the cell is healthy or needs to be eliminated. Now let's look at the different classes of MHC, starting with MHC class I, presenting to cytotoxic T cells. MHC class I molecules are found on the surface of all nucleated cells in the body. Their primary function is to present endogenous antigens, proteins that are synthesized within the cell, including the cell's own proteins as well as those produced by viruses or mutated cancerous cells. Here again we have antigen processing. Inside the cells, proteins are constantly being degraded into small peptides by a complex of enzymes called the proteasome. These peptides are then transported into the endoplasmatic reticulum, where they bind to the MHC class I molecules. Once loaded with peptide, the MHC class I molecules are transported to the cell's surface. Then we have the antigen presentation. At the cell surface, the MHC class I peptide complex is recognized by CD8 positive cytotoxic T cells. If the peptide is identified as self, meaning stemming naturally from the body cell itself, nothing happens. However, if the protein is recognized as foreign, such as a viral peptide or cancerous tumor antigen, the cytotoxic T cell is activated to kill the infected or malignant cell, preventing the spread of the virus or cancer. MHC class I molecules are crucial for the immune system's ability to monitor cells for signs of infection or abnormality, such as in the case of cancer. By displaying fragments of intracellular proteins on the cell's surface, MHC class I molecules enable cytotoxic T cells to detect and eliminate cells that are harboring infections or have become malignant. Then we have MHC class II molecules, presenting to helper T cells. MHC class II molecules are found primarily on antigen-presenting cells or APCs such as dendritic cells, macrophages and B cells. These molecules are responsible for presenting exogenous antigens, so proteins that originate outside of the cell, such as those from bacteria, fungi or other pathogens. Also here we have the classical processes, but a little bit different. Let's start with the antigen processing. Exogenous antigens are taken up by APCs through processes such as phagocytosis or endocytosis. Inside the cell, these antigens are broken down into peptides within the endosome. MHC class II molecules are synthesized in the endoplasmatic reticulum and transported to the endosome, where they bind to the antigenic peptides. Then follows the antigen presentation. The MHC class II peptide complex is then transported to the cell surface, where it is recognized by CD4 positive helper T cells. If the peptide is recognized as foreign, so indicating that the APC encountered a potential threat, the helper T cell becomes activated and begins to secrete cytokines that coordinate the immune response, helping to activate other immune cells, including cytotoxic T cells and B cells. MHC class II molecules are essential for initiating and regulating immune responses, particularly in the context of infections by extracellular pathogens. By presenting antigens to helper T cells, MHC class II molecules play a central role in orchestrating the adaptive immune responses. One of the most remarkable features of MHC molecules is their extreme polymorphism. There are hundreds of different alleles, which means variants, of MHC class I and class II genes in the human population, making it highly unlikely that two individuals, except of course identical twins, will have the same exact set of MHC molecules. This diversity is absolutely essential because of the almost infinite variety of foreign antigens that need to be presented and recognized. Different alleles allow individuals to present a wide array of peptides, ensuring that pathogens cannot easily escape detection by the immune system. This evolutionary advantage has been critical for species survival. However, the same diversity poses challenges in clinical contexts like organ transplantation, where mismatched MHC molecules between donor and recipient can result in the recipient's immune system rejecting the organ as foreign. Consequently, let's look at MHC and diseases. The role of MHC in disease is a topic of significant research interest. Certain MHC alleles are associated with an increased risk of autoimmune diseases, where the immune system mistakenly recognizes self-antigens as foreign and then targets the body's own tissues. For example, the MHC class I allele HLA-B27 is strongly associated with ankylosing spondylitis, a type of inflammatory arthritis. Other autoimmune diseases, such as type 1 diabetes, multiple sclerosis, and celiac disease, also have strong links to specific MHC alleles. In infectious diseases, MHC diversity also plays a critical role. Individuals with certain alleles may present pathogen-derived peptides more effectively, leading to a stronger immune response and better protection. For instance, certain MHC class I alleles are associated with better control of HIV infection, slowing disease progression, while others may lead individuals more susceptible to severe malaria. Interestingly, during the plague or Black Death in medieval Europe, the immense death toll likely shaped the genetic makeup of European populations, including immune-related genes like those in the MHC complex. While it remains uncertain whether specific MHC alleles provided resistance to the plague, the selective pressure from such pandemics likely contributed to the remarkable diversity of MHC alleles we see in the humans today, an evolutionary legacy of survival through immense challenges. MHC molecules are also pivotal in cancer immunotherapy. Tumors often evade immune detection by downregulating MHC class I expression or producing antigens that mimic self. Therapies like immune checkpoint inhibitors, targeting for example PD-1, PD-L1 interactions, enhance the ability of T cells to recognize and attack tumor cells presenting antigens via MHC molecules. These therapies essentially release the brakes on T cells, allowing them to target cancer more aggressively. For example, they have revolutionized the treatment of melanoma, lung cancer, and other tumors, often leading to long-lasting remission in some patients. To conclude, the major histocompatibility complex MHC is a cornerstone of the adaptive immune response, enabling the immune system to detect and respond to a vast array of pathogens and abnormal cells. The ability of MHC molecules to present antigens to T cells is critical for the activation of the immune response and the maintenance of immune surveillance. In our next chapter, we will explore the life and work of Paul Ehrlich, an immunology pioneer known for his side chain theory, which laid the groundwork for our understanding of how antibodies function. Join as we continue to uncover the rich history and cutting-edge science of immunology. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time and thank you for listening.